Good morning, everyone, and thank you for attending today's session. My name is Vicki Lightbound. I'm the Director for the Water Innovation Program here at Alberta Innovates. Before we begin, let's cover a few housekeeping items. At the bottom of your screen are engagement tools you can use. All the engagement tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right corner. If you have any questions during the webcast, you can submit through the Q&A tool. We will try to answer as many of these questions as we can at the end of the webcast. The group chat box is a useful tool to connect with other participants. I'd encourage you to use the tool to discuss the session topic, but please don't let it distract you from the presentation. For the best viewing experience, we recommend closing any programs or browser sessions running in the background that could cause issues. You can find additional answers to some common technical questions located in the help tool at the bottom of your screen. With that, I'd like to welcome everyone to the Alberta Innovates Water Innovation Connect series, Municipal Water Management in a Changing World. Water treatment plants have a critical role in keeping our communities healthy and safe by providing clean drinking water and removing our sanitary waste. As the world changes, whether that means climate change, evolving pathogens, or something else, these treatment plants need to adapt or risk impacting the quality of life of our population. Today, you'll hear from Monica Emleko with the University of Waterloo and Neumann with the University of Alberta. Their bios are available in your engagement tools. For those of you not familiar, Alberta Innovates is a provincial corporation created to support research and innovation activities. We provide funding programs, advice, connections, technical expertise, and applied research services. Our scope encompasses the whole innovation journey, from generation of ideas through applied testing and validation to commercialization and end use. The Water Innovation Connect series will share ideas and outcomes from projects funded through Alberta Innovates Water Innovation Program. The Water Innovation Program is designed to help achieve the goals of Alberta's Water for Life strategy and Alberta's Water Research and Innovation strategy. The knowledge and technologies developed in this program will help to create a clean tech industry in water treatment, support improvement in water use conservation, efficiency, and productivity, and provide safe, secure, and reliable water resources for a growing population while maintaining the health of an aquatic ecosystem. I hope this series will provide you with some valuable information and spark discussion. Now, let's get started. I'll pass it over to Monica, who will be speaking on her project, Wildfire and Pathogen Threats to Water in Alberta.
Thanks, Vicki. I am so very pleased to have the opportunity to speak here on two topics that I believe are really critical to the protection of public health from waterborne disease through the provision of safe drinking water. This work, as you can see on the slide, represents the effort of many colleagues and graduate and undergraduate students over a good few years. And I'd like to acknowledge my co-authors, my colleagues from the University of Waterloo and the University of Alberta who are listed here on this title slide. I'd also like to take a moment to speak about the assigned title of this presentation and of the project, because on the surface, it might appear as a bit unusual of a combination. And so I'll start off by saying that I think of myself as a public health engineer. And I hope that in the next 30 minutes or so, not only will I be able to explain a little bit about what I mean by that, but also I hope that I'll be able to convince you that a talk about wildfire and pathogens as key threats to water, especially the provision of safe drinking water, it really speaks to critical threats to water security, certainly in Alberta, but not only in Alberta, but other regions. While pathogens are universal threats, we can learn a lot from wildfire and its impacts on water security and apply that information to other regions, even if they don't experience wildfire. And maybe they experience climate change exacerbated landscape disturbances such as floods or hurricanes, but information about wildfire is still useful. So let's go ahead and get started. So one important lens through which we consider the provision of safe water is through direct connection to the protection of public health. Now more than ever, in recent years anyway, the COVID-19 global pandemic has emphasized the importance of basic hygiene and sanitation in protection of our health from the spread of infectious disease. And it has emphasized the importance of access to adequate amounts of safe water. Most people think of that as consumption of safe water, drinking water, but there's also a hygiene element, especially hand washing, which we've all heard a lot about recently. So this figure demonstrates that importance. This is from our neighboring United States infectious disease death rates, crude death rates from 1900 to 2000. And the point that it makes here is that with the first continuous municipal use of chlorine in the early 1900s. And that application in water, literally, we saw changes in death rates. And more specifically, if you looked at the major causes of death, the importance of adequate amounts of safe water cannot be emphasized more. Because if you look at these data of the major causes of death during that same period, and then again later on, so 1900 versus 2010, look at that top row of infectious disease. What we see is literally infectious diseases decreased by over 3,000%. So what literally happened was 4 of the 10 major causes of death in the US changed because of access to safe water and sanitation practices. So this is really impressive, and we've come a long way, but we still have a long way to go because the world we live in and how we interact with it is getting more complicated. And so we can think about globally, access to safe water and adequate sanitation still threatens public health. And most people think of it in this global way of not having safe water to consume. But it's important to emphasize that the most vulnerable populations are the ones who are hit the most severely. The figure on the right here indicates that simply diarrhea affects young children under the age of 5 as a major cause of death, outpacing illnesses such as malaria and AIDS. But for us in Canada and in Alberta, it's also critical to recognize that this isn't just an international concept. So I pulled from yesterday. So yesterday, November 17th, major water advisories across the country. And yes, Alberta's faring well, so that's good. But still, we can do better. We have water advisories across the country. And people ask frequently, you know, why do we have water advisories? Why do they occur? Why is it okay? We have boil water advisories. We have do not consume advisories. Not so much now, but in the warmer months, we have cyanobacterial blooms or algal blooms. Sometimes we have water shortages. By far, across the board now, the big issue is boil water advisories. And it's really important to understand that they may be called because of perceptions or realities of contamination. They may be called for operational reasons that don't necessarily mean there's a health threat. And it's important to know that we keep water flowing because of operational reasons. Yes, we want to be able to wash our hands, and that's important. That's one reason. But another reason is because if we shut down plants, it's a lot of work and effort to get them started up. And so the issue of water advisories is complicated, but a key message that comes from understanding them is that we have to keep water flowing in order to protect public health. And when we talk about water, we need to think about the contaminants in that water. People often think about either pathogens, so microbial contaminants, or chemical contaminants typically that are drivers of toxicity. And so when we think about those from a public health perspective, typically what we're thinking about is acute risks versus chronic risks. And so it's not surprising that the pathogens, the acute risks, form the primary objective of water treatment. We're looking to manage those risks. So this couldn't be more clear in statements that have been made by the World Health Organization that has emphasized, for example, that we use disinfectants to treat microorganisms. However, disinfectants can react with other materials in water and produce disinfection byproducts, which are also regulated and may be contributors over the longer term to chronic illnesses, such as maybe even cancers, although that discussion is still out as the toxicology is not well understood. So the World Health Organization has emphasized that we need to focus on pathogens because the risk of death from pathogens is at least 100 to 1,000 times greater than the risk of cancer from disinfection byproducts, and the risk of illness from pathogens is at least 10,000 to 1 million times greater than the risk of cancer from disinfection byproducts. So it's critical to remember that treatment of pathogens is the paramount objective of water treatment, and this is how we go about protecting public health. So how do we achieve that? So I mentioned this idea of I see myself as a public health engineer. So, my job is to make sure that we can actually treat the water that's going to result in public health protection. So let's talk about treatment. Let's talk about treatability of water. Most people are very surprised to find that there are only a few key drivers of infrastructure design and operational challenges that can really get us most of the way, not all of the way, but most of the way to protecting public health from waterborne disease. And notice these parameters on here, they're turbidity, so suspended solids, and color and total organic carbon, which are both related to natural organic matter or dissolved carbon present in water. Interestingly, none of these parameters are directly, or neither I should say, are directly linked to public health protection from a health perspective, right? They're not contaminants of health concern. What they are is contaminants that drive what infrastructure we need to treat water and how easy or difficult it will be to treat that water. Now, we can treat water on the space station. We can treat water in most environments. The question isn't, can we do it? It's how much is it going to cost? That's the big challenge. So when we think about the basics of water treatment, it is not plug and play. We have to design for water quality. And without getting into the details of all of these processes presented here, all of that big infrastructure is focused on removal of solids or turbidity and natural organic matter, so that TOC or DOC, so total or dissolved organic carbon, which includes color. And then we do that so that we can effectively treat the pathogens, and we're good at treating pathogens. But what's happening is climate change is resulting in conditions that make that harder. And why is that? It's because water quality is changing, and it effectively is a moving target. Water quality can be extremely deteriorated, and we can treat it. What makes it difficult is when it fluctuates and fluctuates over short periods of time. So whether it be fires, floods, hurricanes, if they're significant enough to affect source water quality, it means having more challenges in water treatment. And it's kind of like the analogy that I would give is if you have a snowmelt freshet that you're really challenged in treating because water quality changes so quickly, imagine having several of those over the course of the year instead of one. So that's the kind of situation that we're potentially facing in some cases with respect to climate change, exacerbated landscape disturbances that affect our source water quality. So then, okay, so how do these disturbances actually affect these parameters that affect water quality? So let's look at that. And let's look at it through the lens of fire. So first and foremost, in Alberta, let's be clear about the situation. So this left figure talks about how much of the landscape is burning. So it's annual area burned. And what we're seeing is that the area burned is changing. And specifically what's happening is that when we look at the different sizes of fires, and that's the middle schematic, and again, the percentage of the total fires, what you see is the biggest fires are a very small fraction. Greater than 90% of fires are less than 1 hectare in size. Less than 0.3% of fires are the big fires that result in more than 98% of the area burned. And why is that? That goes to the last figure. To have a fire, you need the right conditions. You need fuel on the landscape. The relief on the landscape, the topography is going to affect that, and weather is going to affect that. So with changing climate, we're seeing more severe burning conditions, longer fire seasons, more severe fires. And does that affect water quality? Yes, it does. Wildfires can significantly impact many aspects of water quality. And what you see in this image here is not only can they affect water quickly, like a debris flow that you see in the bottom left corner, that's more relevant to environments like Colorado. Doesn't mean it won't happen in Western Canada, it's just more relevant. In Western Canada and parts of Alberta, what we've seen, whether it be in the montane cordillera of the Southwest or if it's in the boreal plains, what we see is a more prolonged effect. Not everything comes downstream, but rather it comes downstream in pulses and it persists. So it's important to understand the environment and the setting that we're talking about, because Alberta is not Colorado, and that goes for other parts of Canada as well. And so when we look at fire as one example of a disturbance, the leftmost picture here is what some of our study systems have looked like previously. And then most people, the second photo from the left is what most people imagine, this surge of black water coming through the system. But what I'd like to suggest is that these impacts can actually linger for decades, and they can have cascading impacts on both ecosystems and water quality in those ecosystems. And so therefore, they're going to have impacts for water treatability and public health protection. So not only can we expect these impacts we've seen in high quality systems that have the most to lose, but even environments like we've seen in Fort McMurray in the regional municipality of Wood Buffalo after the 2016 Horse River fire. That was already a challenged system. And yet after the fire, we saw very real impacts in Fort McMurray, where we saw increases of 50% or more in chemical coagulant demands, and the occurrence of annual potentially toxin-forming algal blooms in the source water that they have not experienced regularly. And you can see some of the images on the right, literally the tanks looking green because of those algal blooms. So these challenges can threaten public health if they're not adequately managed. And fortunately, we've been able to do that in Fort McMurray, but there's a cost to it. And so let's step back and more broadly look at these issues. So these data are from the Southern Rockies Watershed Project, over 15 years now of looking at landscape disturbance impacts, specifically wildfire impacts on a range of water issues in southwestern Alberta. So this is after the Lost Creek fire. We've seen, if you look at these figures, we've got sediment production or concentration and turbidity. Black is reference unburned catchments. Green rather. Black is burned catchments, and the brown or orangey brown is post-fire salvage log catchments over now a decade. And what we've seen is there are wet years and there are dry years. There are bigger differences in some years between the burned and impacted catchments versus the reference catchments. And some years the differences are fewer, but they're there and they persist. And if you look at this photo, you can see why with the figure below it. Our glacial history, our surficial geology has led to conditions that lead to a tremendous amount of sediment on the landscape, and it persists and leads to these water quality impacts. Similarly, we have seen long-term impacts in nutrients like phosphorus and the algal growth or proliferation that it promotes. And again, they've persisted over decades and can be expected to persist because these materials stay in our systems. And it doesn't mean that we go from crystal clear water to black water, but rather that water quality is fluctuating and typically in conjunction with high flow conditions. Now, we have nitrogen here at the bottom of the screen to show that not all aspects of water quality continue to fluctuate over time. Some come down to baseline or recover after a few years' time. But of course, we talk about in the water industry, rapid recovery on the landscape might be a few years, but in a water treatment plant, it's a few hours. So the timescales are different. And so this figure here that focuses on dissolved organic carbon, that natural organic matter that contributes to significant challenges in coagulant consumption and costs, we can see here in those same watersheds that we've studied that there continue to be spikier and more spikes after wildfire. And that is very analogous to what's been experienced in Fort McMurray, which we can show as strongly as we possibly can, that there is a linkage to the fire with respect to what's being observed in the water plant, with respect to coagulant costs, and that impact is persisting to this day. Very, very, very clearly connected. And so these are some of the challenges that we need to think about as we prepare to move forward in a climate that is changing. So on this slide, we have listed several wildfire threats to drinking water treatability that are related to, on the top row, the aspects of water quality that change after disturbance. We focused on wildfire, but it's important to note that this table is also relevant to not just wildfire, but also other disturbances. And again, going back to the schematic that I showed previously, it's because of impacts on the upfront treatment. Solids, turbidity, and carbon removal prevent us from being able to treat pathogens well if we don't remove them adequately first. So what do we do? how do we manage these risks to public health that are posed by potential threats to the provision of safe drinking water? So let's examine the extremes. One of the things we did in our project was we looked at, okay, we have various water qualities prior to fire. Let's look at the most extreme case, these episodic, short-lived black water events. Sometimes they don't even happen after fire. But the idea was, if we can treat this, well, then we can treat water that's less degraded. And the thing with water treatment that's important to know is that we're not always equipped to respond immediately to these challenges. And so we decided to do some work creating these scenarios so that we could actually thoroughly investigate them. So on the landscape, we went and collected not Ziploc bags of ash, but in partnership with our colleagues from British Columbia, hundreds of 5-gallon pails of ash. So wonderful partnerships enabled us to do that. And then we went about creating reproducibly black water matrices that would literally represent the most severe deterioration of ash running in from the landscape and yielding those changes not only in turbidity that you can see visually, but also in dissolved organic carbon, and doing so reproducibly. And we conducted first bench scale tests of various treatment technologies that we thought should be able to deal with some of these challenges. So not only did we look at conventional treatment, but we also looked at ballasted sand flocculation, which is a technology that is specifically designed to be responsive to rapid changes in water quality, especially with respect to changes in solids loads. And these ballasted sand flocculation, ActiFlow is an example of that type of process. And we see these processes across Alberta. And so this is what we tested at bench scale at a variety of conditions. And I should point out, we did this in partnership with the City of Calgary using the Elbow River Water Matrix. We also did subsequent work in partnership with the regional municipality of Wood Buffalo, and looked at all sorts of pretreatment chemicals and conditions and found basically the same results regardless of the chemicals. So it wasn't that you're going to find a magic chemical that's going to be the silver bullet in treating these, but rather it's more about the treatment philosophy and the technology approach. And so what did that look like? Quite simply, with a range of these technologies, so specifically we looked at conventional treatment, which is coagulation, flocculation, clarification, usually by sedimentation, this ballasted sand flocculation, and then we also looked at ballasted sand flocculation with enhanced coagulation. Why? Because that helps us in dropping the pH of the water so that we can precipitate solids. And the long and short, as you can see here on the left, is most of the technologies removed turbidity very well, but carbon, the NOM, was a challenge. So we adjusted. That's what treatment engineers do. And so we said, okay, let's go to the next level. And although enhanced coagulation is supposed to be a best available technology, we can explain why that didn't work as well. When we included the addition of powdered activated carbon with ballasted sand flocculation, we got to the point of being compliant with respect to treated water targets. Now remember, when I say treated water targets here, I mean water that would enter filters, not water that we would consume, because there's subsequent treatment in this treatment process, as we saw in our schematic. So operational targets here, not consumption targets with the water. So this was a great success. But of course, in water treatment, we have to pilot things. And so we went on, and this project was fantastic. What an amazing opportunity to be literally the first group globally to be able to test the treatment of severely deteriorated black water after fire at pilot scale. This literally took hundreds of pails of ash. Those are 5,000-liter tanks that you see up there. This is a dream PhD project for the student who worked in partnership with the City of Calgary, Jesse Swark, one of the co-authors, and also with Veolia Canada. And we tested ballasted sand flocculation with powdered activated carbon not that long ago. You can see the black water in here. And the long and short is that we could very easily see what we expected, frankly, which is that extreme water requires extreme treatment. But ballasted sand flocculation with powder activated carbon, you can see the results for yourself, ranging from the black water to the pass of treatment after ballasted sand flocculation to then after powdered activated carbon. And these are off-the-shelf technologies, by the way. It's just a question of having access to be able to set up and implement them, which not all utilities have. And so I want to point out, we're not suggesting that everybody who's in a fire-prone region invest in these technologies, but what we are suggesting is that we need to talk about, is it worth investing in this direction, or are there other things that we can do to be more operationally resilient? because the probability of seeing these black water events is not that high. And even when they occur, they typically last for a very short period of time. So having dealt with the issue to some extent, feeling some success with respect to treating water post-fire, now let's talk about pathogens. Now the thing with pathogens, because we've got the upstream treatment, let's focus on downstream. The thing with pathogens is that pathogens are pretty easy to treat in that we know how to do it. And so usually the challenge is as long as we can get everything upstream to work, we'll know what to do downstream. The more current challenges with pathogens have to do with how do we speak to how much treatment do we need? So in other words, risk assessment. So let's take the treatment of viruses in groundwater as an example. Our Canadian context from Health Canada's guidance document on water quality and risk assessment speaks to a health-based treatment target. This is the guidelines for Canadian water quality, specifically a health-based target that is a treatment goal of a minimum of 4 log removal and or inactivation of enteric viruses. Okay, in contrast, our neighbors to the south have a very different approach, where they include 4 log treatment for viruses if the water has attributes of surface water. But in cases where that is not the case, there's a belief that maybe we don't need any disinfection. Aren't subsurface water supplies pristine? And to the microbiologist, most waters are not pristine because animals use them to bathe in, and water from the surface makes its way to the subsurface. And so this is one of the big questions we set out to ask, because we, more specifically, we're saying, okay, is this across the board 4 log treatment requirement too much? Is it too stringent? Do regulatory frameworks with lower treatment requirements provide adequate treatment and public health protection from viruses? And of course, in the background, we're thinking, well, bacteria are there too, but bacteria are easier to disinfect than viruses. And so our focus tends to be viruses. And then if we need even additional treatment, the less commonly occurring in the subsurface protozoa come into play. But here we're just focusing on viruses. And so the approach we use is quantitative microbial risk assessment. QMRA is actually a set of approaches and tools that integrates information about pathogen concentrations in the raw water source, our water treatment capabilities, the consumption of water, and then dose-response relationships to evaluate risk and the possibility of becoming ill if we consume this water. And it's not that there should be none of these pathogens in the water, but all of these frameworks assume some level of acceptable risk. So it's important to recognize that this process of QMRA can be turned around or reversed so that a required level of water treatment can be determined given an acceptable level of risk. So let's look at this in this context. So here we used norovirus as occurrence data as the virus we were analyzing, because we had access to a community intervention trial that actually occurred in the United States with our colleague Mark Borchardt in Wisconsin. And that trial was exploring whether or not implementation of UV disinfection affected rates of acute gastrointestinal illness. And so we were using qPCR data here, and it's important to note that of 392 samples, 360 were non-detects. And so we were looking at average concentrations of 3.8 genomic equivalent copies per liter to somewhere in the vicinity of a maximum of about 294 equivalent copies per liter. And based on assumptions of consumption of unboiled tap water and these data, we looked at, okay, what level of treatment would be required? And so you can see here, we've zoomed in on this axis, and you can see that a very small fraction of these well samples has certain concentrations of norovirus in there. And what we did was we looked at the literature. People tend to think risk assessment is this black box accepted approach, but of course it's still in the domain of research and assumptions. And so we looked at various models for dose response. So if you consume some of the virus or the water containing the virus, what might happen to you with respect to illness. And you can see here in the blue table that host susceptibility, immunity, and aggregation of viruses in these water supplies are all key factors that can play into dose response. And so when you look at these various models, you can see that there are 2 basic clusters. The first cluster is around a high probability of infection at low doses. and is what is commonly used in the literature and a lot of regulations. The second model, however, the second cluster of models has better empirical fit to our data sets that we've seen. And so we said, okay, let's look at all of these. Let's not get into my model is better than your model, but let's look at all of these and see what they mean for risk. And so interestingly, when we looked at these results, and it's important to point out that these results were from what was deemed to be and expected to be safe groundwater. The deep, very deep wells where we really didn't think viruses, or some people anyway, didn't think viruses would be present. But of course, Mark Borchardt has been going on for years about that, in fact, there is risk present in these systems. So not surprisingly, when we went through the risk assessment, for each sample, we calculated how much treatment would be needed if the concentrations were sustained year-round that were observed in that system in Wisconsin. And with this approach, most of the concentrations at which norovirus was detected would require, in fact, a minimum 4-log treatment consistent with Health Canada's guidelines and the guidelines that are used in Alberta. And you'll note here in this figure that the use of the more extreme dose response models that don't actually fit the data added up to a 3-log calculated increase in treatment needs. And so we then looked at this more specifically and evaluated treatment targets based upon some of the summary statistics from the data. So we looked at average reductions required. That's how our regulations are set. But then we look at the more extremes, like the maximum concentrations. And again, what you see is that depending on which dose response model you use, you can have these shifts. But overall, we thought that this work quite clearly showed that a 4-log treatment goal is warranted. We can get into why these models that don't fit don't make sense to apply, and that we can use our scarce public health protection dollars in better ways to protect public health than by requiring more treatment of viruses that would be unwarranted for some supplies. So we can feel, the net result is we can feel pretty good about what we're doing. Another project that we've been involved in is one that's really interesting because it has to do with high school mathematics and how we report data. And really what's been done across the board in water policy and beyond, it makes its way into the food industry. And so the concept is a pretty straightforward one. You can't be in the industry, water industry, and not talk about average or log removals, average performance and log removals if you're interested in public health. And we talk about log removals because of the desire to have a very high rate of inactivation of pathogens. So 1 log removal is 90% removal or disinfection, 2 log is 99%, 3 log is 99.9%, et cetera. And you see this in commercials for hand sanitizers these days and for other surface cleaners. And so the issue that came up here is one that one would deal with in high school, literally. It's that if I take the percentage removals and I average them and I take the log, so 99.8, and I retain all those decimals, 99.999, if I take the average of those and then take the logarithm, that's not the same as calculating the log of each of those percentages and then calculating the average values of the logs themselves. And this becomes really a very important point of confusion because a lot of people take the average of the logs. And our position is that that is wrong. And let's talk about why that is. So first and foremost, high average log reduction does not necessarily correspond, as you see in this figure, to higher average percent reduction. So you can see the red distribution and the blue distribution, 2 treatment processes. Which one is more effective at treating and protecting public health? If you calculate an average log, you can see that it looks like the red distribution, the red technology, treatment technology B, is more effective than treatment technology A. But if you average the percent reductions and then express that as a log value, in fact, the blue technology, treatment technology A, is more effective. And again, it's simple mathematics. And so these outcomes both can't be right. One should be accurate. Let's walk through an example to look at that. And the example I'd like to walk through has significant consequences because it is this type of error is made in the microorganism elimination credits that have been proposed by the World Health Organization and by colleagues from the Netherlands who reported these data that then were picked up by the World Health Organization and implemented not only in the risk assessment guidelines that they provided, but also in other guidelines such as those that Health Canada uses. And so there's a table in some of the work that we've done that actually points to those connections from those documents and then connects it back to our policies. And so the best way to look at this impact is through an example. So very quickly, I just like to assume we have, let's assume that we have two populations and explore the implications of misusing average log reduction values upon risk. So we have these two cities, each with a population of 100,000 in our figure, a raw water supply with 1 oocyst of cryptosporidium, so that's a protozoan pathogen, per 100 liters. And let's assume they apply the same treatment technology, but for whatever reason, water matrix, design, operation, one city achieves only a 2-log treatment on the left, and the other achieves a 4-log treatment on the right. Now, without additional testing, neither utility knows how effective its particular implementation of the treatment technology is. So this is why we have it here in gray. And so now suppose that we believe, perhaps from these tables in the literature, that the average treatment that we should expect from our well-operated treatment technology is 3 log. That's the average of the 2 and the 4. If I average 2 and 4, I get 3. Okay. And so then when I walk through these progressions, we would think that we would get 10 infections per year in each city. However, the first city has 1 log less treatment and more infections, while the second has 1 log more treatment and fewer infections. So in the first city, we would see 100 infections. In the second, we would see 1, and we would expect the total to add up to 101, but in fact, it only adds up to 20 because of the 3 log. In contrast, if we do the math the other way, where we're now taking the log of the averages, we again go through this same process and we see a different outcome, which is that our effective log reduction, this is a phrase that we've suggested based on this work to emphasize that it's not an average of the logs. The effective log reduction is 2.3. And then when we go through these calculations again, we do in fact get the correct estimate of the number of infections in the 2 cities, and it adds up to 101. So although the risk is not correctly evaluated, both cities could benefit from some site-specific performance monitoring, because this is just an assumption based on table values, right? But at least it comes closer to giving us a proper average based on the data that are available in the literature. Of course, site-specific monitoring would be useful, but it does emphasize that when we look at, nonetheless, the guidance and the regulatory process, if you look now at quantitative microbial risk assessment in the World Health Organization document, there's an illustrative example there. And the guidance that is given in that document using an average log reduction, so that's the dotted line in red, versus the arithmetic mean risk, which is calculated with the effective log reduction, we see a difference. And the difference is not trivial. So when we reanalyzed this log reduction value and concluded that this risk was actually higher than what one might have expected otherwise. So in other words, there would be a greater treatment risk. And what we're seeing here is the math works out, one is an arithmetic mean and one is a geometric mean. And the question is, which one is appropriate? Now, the calculation should be done appropriately from a mathematical standpoint without a doubt. But the key thing to note is, even though we have examples like this that have this overlooked statistical error or calculation error that results here, as we've shown, in almost a 1 log difference, it's a 0.9 log difference, and potentially an incorrect conclusion about which treatment technology outperforms another one. It's important to note that while we have these approaches, not only in the World Health Organization document, but also in Health Canada's guidance, it's not like they're resulting in massive outbreaks of disease. I think we need to recalibrate and think about setting targets and making sure they're adequately reflective of the data that we have. So it looks like we have to do a lot of re-examination of data. So these are the ways in which we can act to protect public health on the pathogen front, to really be rigorous in our risk assessment and our quantitation of risk, and most importantly, to speak about the fact that it's an evolving process. This is not a black box plug and play kind of thing. We need to have open discussions about the models and link them to data so that we can be most protective of public health. So in conclusion, I'd just like to wrap up to say that there have been great strides in the protection of public health through the provision of safe drinking water, but there still is a road ahead of us. Yes, for sure, pathogens in water remain the paramount objective of drinking water treatment, and we have to recognize that climate change exacerbated landscape disturbances can threaten that treatment and public health protection, very likely in an episodic kind of way, because in impacting upstream treatment, they can prevent adequate treatment of waterborne pathogens and therefore limit how much water we can provide, so potentially lead to service disruptions or alternatively complete outages. However, there's hope on the treatment side. We've shown that severely deteriorated water can be treated, and that means less deteriorated water can be treated, and we need to have discussions about what's going to be operationally significant with respect to managing that water and the associated risks, because the extremes are not likely. It's the less deteriorated water that we need to be in a position to manage. And we have to ask ourselves, is it worth the cost? How can we best be efficient with protection of public health through the provision of safe water? And part of that includes managing how we report microbial health risks and how we then include those representations and models to set regulations. And we've shown some examples of being in a really good place with respect to requiring disinfection, but also wanting to take a look at some of those values to make sure that we're calculating them as well as possible so that we can do the best job in evaluating the efficacy of the treatments that we recommend. And so this is going to be relevant now and in the future as we look to not only the issues that are known challenges, but to emerging issues. And I'm looking forward to hearing more about emerging treatment challenges and public health protection issues from Norm Neumann. So thank you very much.
Thank you, Monica. Some great insights on our changing water inputs and changing understanding of our treatment process. Reminder to continue to submit your questions for both speakers throughout the session. We'd love to have a great discussion with you, whether it's on specifics of the presentation or your general, hoping to get general thoughts from the presenters on the topic. So with that, I'm going to pass it over to Norm, who'll be speaking on his project, Evidence for the Evolution of Water Treatment-Resistant Pathogenic C. coli, our favorite speaker to line up right before lunch.
And great, thanks, Vicky, for that. I want to start by telling you a little bit of a story. And it comes from a colleague of mine who was in his undergraduate program in engineering at a university, and he was in a class on water treatment. And at the end of that class, he asked a question to the professor, and he said, "Do you think microbes can evolve resistance to the treatment that we impose on them?" And the professor said to him, "That's something you'll never have to worry about." And I say that story not to criticize the professor, but if I was to look at my own perceptions of water and waterborne pathogens, 3 years ago, I would have said the same thing. And I want to take you on a little bit of a journey of discovery that has, for lack of a better term, rocked my world in my perceptions about this. And I want to share with you with this. And I want to ask a basic, very question. Are we on the cusp of a public health crisis? This funding is provided by Alberta Innovates. I can't emphasize enough how important the funding is that we've received over the years, particularly from Alberta Innovates in the city of Calgary. And the contiguous funding and the investment into the research programs that we have, it's been generous. And hopefully you can see the importance of this research as we walk through this story of discovery. And in order for you to really understand the story, I have to introduce you to one of the actors that we work with. And this is an organism called Escherichia coli, which is a bacteria. And Escherichia coli is a bacteria that was discovered in 1885 and is recognized as a really important indicator of fecal pollution in water for the last 130 years. Its presence and concentrations tell us about the quality of that water, and that can be drinking water, recreational irrigation, or even when we discharge wastewaters. The bacteria itself colonizes the gut of mammals, birds, and reptiles, and is thought to be what we call a generalist. And I mean a generalist is that the E. coli that seems to colonize us as humans seems to also colonize birds and vice versa. And so we think that a multitude of hosts can act, or there's a multitude of animals that can act as hosts for E. coli. This particular organism holds a near sacred status in the field of microbiology. It is the organism that we have studied the most. And we put a lot of faith in our understanding of this organism, and a lot of our interpretation of water safety risk is rested in this single organism. And because so much rests on us understanding, we are often afraid or placed under intense scrutiny when challenging the status of this microbe. And so our journey begins with a theory that we had that challenged the field. And the theory was that, in fact, all E. coli are not created equally. So that the E. coli that colonizes a human is different than an E. coli that colonizes a dog or a cat or a horse or a pig or a cow or whatever. And this has major implications for water safety, because it would suggest that perhaps maybe animal feces is less risky than human feces when it's in water. It suggests that some of the pathogens that we see from animals may not be infectious to humans, and therefore may not be the risk that we think in terms of their zoonotic potential. And we challenge the conception that E. coli is not a coherent species, we said, but rather a species complex, or a collection of genetic types that are so similar that it's difficult to distinguish between them. And what do I mean by that? Well, if I give you this picture, one of the first things you probably recognize is the various colors, and they stand out. You see red, you see violet, you see blue, perhaps teal, green, yellow. And we can think of those as potential individual wavelengths in a spectrum, just like we can think of these as potential individual species in a spectrum. But in between all these different colors is subtle changes in the evolution of that color, and the same thing might occur with E. coli. And that was our theory. And we published a couple of papers back in 2015 and 16 on this theory that provided evidence that this theory was probably correct. And so I'll show a figure from here, or a table from here. So for example, we looked at 105 different human strains, genetic strains of E. coli that came from humans. And in there, 53% of those strains actually possessed a genetic biomarker that was 100% specific to humans, suggesting that out of those 105 strains of E. coli, approximately 58 of them were specific and only found in humans and couldn't be found in any of the other animal species that we see here. If we look at coyotes, for example, as another example, we had 44 different genetic strains of E. coli, and we found that 61% of these particular strains had a genetic biomarker that was 100% specific to coyotes. And these were very much different than those that we've seen in dogs. And this is important because it would suggest that closely related species, in this case two canids, the domestic dog, or Canis domesticus, and the coyote, Canis latrans, that the E. coli that colonizes these very close canid species are actually genetically different. And our intent in doing this was to say, wow, wouldn't it be neat if we were able to test water and not just determine whether E. coli was present, but was to understand where the sources of pollution might be coming from. And if we could identify these genetically as being distinct, perhaps we might have a set of tools that we could use in the lab to determine where the sources of pollution might be impacting water. And we could go in and mitigate those sources. So one of the first things we did was we thought, okay, the simplest way to test this is actually to test it on wastewater. Because when you think about sewage treatment plants, or municipal sewage treatment plants, that it's probably comprised mostly of human wastes, and therefore the E. coli that we would see should have a human signature. And we found a remarkable thing. We found that of the wastewater strains that we had in our library, and there were 68, 82% of those strains carried a biomarker that was 100% specific to wastewater. And our first response was, what? What do you mean? I mean, 99.99% of all the bulk of E. coli that's flowing into a sewage treatment plant is coming from people flushing their toilets. How could this be true that there are E. coli that have nothing to do with the feces that enters into the wastewater treatment plant in the first place? So we've published a number of papers on a phenomenon we call the presence of naturalized wastewater E. coli. And these are very bizarre E. coli. So these strains of E. coli actually appear to have evolved to survive and grow in a wastewater treatment plant. In other words, they've abandoned the life in the gut of an animal or a human, and they've actually evolved to live and survive in sewage and wastewater, is what we think. And this may seem kind of bizarre and crazy, but when you really think about it, a sewage treatment plant is nothing more than a really large digestive tract. And in fact, the E. coli that come out of a wastewater treatment plant, the large proportion of the E. coli that come out of the wastewater treatment plant after treatment are or were represented by these naturalized strains. And a very remarkable finding was that we could find these strains in all wastewater treatment plants in Alberta that we looked at. And not only could we find it in Alberta, but we could find it in wastewater treatment plants in the US and even Europe. And so not only has this organism evolved to live within a wastewater treatment plant, it's spread itself around the entire globe. Extremely successful. And most importantly, they appear to be resistant to wastewater treatment. So we've been actively looking at this phenomena and published a number of papers looking at this whole phenomena of evolved resistance in E. coli. The most recent paper that we've published is just this year, earlier this year, entitled "The Locus of Heat Resistance Confers Resistance to Chlorine and Other Oxidizing Chemicals in Escherichia coli." And I want to show you a particular graph that we've published on in this particular paper, and this was with colleagues from the Faculty of Agriculture, Life and Environmental Sciences. And so if you compare susceptible and resistant strains to E. coli, we find, for example, that the naturalized wastewater E. coli are 100 million times more resistant to heat, 60 degrees Celsius, than susceptible strains. And you can read that on the graph on the right by looking at the red arrow to the right and the strain that's represented in the susceptible group. And if you notice on the y-axis, that's a log scale. And how you read this is that if you had 100 million bacteria of this susceptible strain and you treated it at 60 degrees Celsius for 5 minutes, you would kill all of the bacteria. If you look at the red arrow to the left, if you look at the resistant strain, if you did the same treatment of 60 degrees for 5 minutes, you'd be lucky if you killed 1 out of 100 million. In other words, the naturalized wastewater strains were 100 million times more resistant to heat than the susceptible strains. Another important finding of this was the chlorine resistance. So if you look at the blue arrow on the right, we have a susceptible strain that if you had 1 million of these E. coli and you treated them at chlorine at a specific dose for a certain time, you would kill 1 million, all 1 million of those cells. Whereas if you look at the blue arrow on the left, under the wastewater resistant strains, you'd be lucky if you killed 10, suggesting that naturalized wastewater strains are up to 100,000 times more resistant to chlorine than susceptible strains that we see. In fact, what we've been able to identify from these naturalized treatment-resistant wastewater E. coli is a remarkable finding that virtually anything that we throw at them, they've already developed a strategy of resistance. So I've talked a little bit about chlorine, for example, and that some strains are 100,000 times more resistant to chlorine. Some of these strains are extremely efficient at production of biofilms as well, too. We know that in the case some of these organisms are also resistant to advanced oxidants, including UV. They carry an abundance of UV resistance genes and antioxidant resistant genes, and they're resistant to peroxide and other advanced oxidants that we've tested them against. During wastewater treatment, we alter the nutrients. The nutrient composition within a wastewater treatment plant is a lot different than the gut. And what we find remarkable about these strains is they've acquired metabolic genes from other bacteria that allow them to use the nutrients within a wastewater treatment plant for their growth. And that they've adopted a growth strategy that allows them to grow at low temperature. So typically we think E. coli can grow at 37 degrees in your gut. Well, these can actually grow at 20 degrees and are extremely happy at 20 degrees, even 15 to 20 degrees in their growth. We use such things as microbial competition or predation or infection to destroy these bacteria during a wastewater treatment plant in the process. And the idea is that microbes consume other microbes. So if we create an environment for certain microbes that feed on these, perhaps these will become a food source. And what's remarkable about these strains is they've developed a number of ways to outcompete other bacteria, some of which include things like toxin, antitoxin genes. And toxin antitoxin genes are quite unique because they represent the biowarfare between microbial species. In other words, these microbes are able to produce poisons that poison other bacteria. But while they produce the poison, they also produce an antidote. And as they come in contact with an enemy, they inject the poison into their enemy, and while they're injecting it, they remove the antidote. And the poison gets injected into their enemy, and their enemy dies. And these types of strains are profusely abundant in these toxin antitoxin genes. They're also highly resistant to any kind of viruses that can infect them, and virtually impossible for us in the lab to infect with viruses. Although we don't use antibiotics in treatment, antibiotic concentrations exist within wastewater. And what's remarkable about these naturalized treatment-resistant wastewater is they carry an abundance of antibiotic resistance genes. Sorry, that should be below here. And we've already talked about the fact that they're resistant to heat. And you wonder why in the world would a wastewater E. coli want to be resistant to heat when water is never more than 20 degrees Celsius in a wastewater treatment plant? Well, we have to remember that in a wastewater and a sewage treatment plant, as sewage comes in, it's divided into two phases. It's divided into the liquid phase and the solids phase. And it's the solids phase that then goes on to be composted. And in composting, we rely on heat to destroy the bacteria. And the US EPA recommends that we should use 55 degrees Celsius for 3 days to inactivate all the E. coli within a sample, within a compost heap. Well, as I've already told you, these things are 100 million times more resistant to heat. In fact, we've been able to find E. coli that can survive 60 degrees Celsius for 8 days in the lab. So for everything we throw at them, they have already developed a remarkable adaptations to survive, which is astounding. Monica's talked a little bit about microbial risk assessment, and this becomes an important element as we try to understand resistance, because when we look at treatment, we often use those organisms that we think are the most resistant to a particular treatment and use them as a surrogate to understand treatment. So Cryptosporidium and Giardia are surrogate organisms for chlorine, as well as advanced oxidation. And things such as adenovirus are those that we use to set disinfection criteria for ultraviolet light. And it's generally assumed that bacteria aren't as resistant to these treatments, so we don't really worry about them. And the idea is if we can treat water to the level to control these surrogate organisms, then surely the bacteria will also be controlled. And this is unfortunate because literally in all inactivation studies that are carried out in the lab in support of legislation and regulations, we use susceptible strains of E. coli. So we use these strains to evaluate log reductions for E. coli. We don't use these strains. And this is quite important because one of the concepts could mean that could some E. coli strains actually be more resistant to chlorine than Giardia or Cryptosporidium? And for you as engineers out there, or even environmental microbiologists, this is heretical. To even say this is probably sacrilegious, and something that 3 years ago I would have said was heretical and sacrilegious in the whole area of environmental microbiology. But is it? We've known about the problem for quite a while. There's a paper published by an Israeli scientist back in 1964 who described the phenomenon of chlorine-resistant bacteria and the need to have a multi-barrier approach. In his paper, he quotes two other papers, one in Russian, and that one I haven't read because I can't speak Russian, but was published in 1947 describing the secondary appearance of Bacillus coli or E. coli in chlorinated water. And another paper back in 1949 that described the whole phenomena of chlorine resistance. So we've known about it for a long time, but is it something that the scientific community has largely ignored because we thought that bacteria could never develop this level of resistance? And so part of our work really looks at the sanitation process, and we took a different approach to understanding the sanitation process. So you see on the left-hand side is raw sewage comes in. As you flush your toilets and all that water comes into the sewage treatment plant. When it comes into the sewage treatment plant, you'll have a concentration of E. coli of about 1 million E. coli per 100 mLs of water. And we put this through a series of different treatments that are there. And the final discharged effluent is typically around 100 E. coli. So we move from about 1 million E. coli to 100 E. coli, and we have a 4-log reduction across that process, which, as Monica's talked about, is equivalent to 99.99% reduction in E. coli. And I call this the great microbial Powerball lottery. It's luck. We think it's luck that they survived, that the 100 survived because they were just lucky as they got through the system. But when you really begin to understand that these sanitation processes that we use also represent strong natural selection forces that might be driving the evolution, we asked a very different question. And we asked this question: why did the 100 E. coli survive and the other 999,900 die? Was it really luck? And what I can tell you is this, and what you've already seen from the data that I presented, is that inactivation or killing of microbes is not random during the sanitation process or the sewage treatment process. The 100 E. coli survived not because they were lucky, but because they had what I would call microbial Kevlar. In other words, they were wearing bulletproof vests. And the very interesting thing about this whole thing is it's genetically encoded, suggesting that they pass it on to their progeny, this Kevlar vest. And so one of the questions that we really do when we have these 100 E. coli at the end of our wastewater treatment plant is really, are these microbes pathogenic? And in some ways, you can already begin to answer that. And part of the answer is no, because as I've already told you, the naturalized wastewater E. coli appear to have abandoned life in the gut of an animal or a human for life in the wastewater treatment plant. They just happen to be so adapted to this that's where they survive. But that doesn't mean they're not a risk. We know that because they've developed these mechanisms, they may act as a reservoir of water treatment resistance genes for other pathogens. They may be a reservoir for antibiotic resistance genes. And they may be a reservoir of genes important for environmental persistence, allowing them to survive longer in the environment. The other important thing that I've already mentioned to you, though, is that about 50% of the E. coli that we see about on average coming out of a wastewater treatment plant is comprised of this 50% naturalized wastewater E. coli. That still leaves the question, what about the other 50% that come out? Could these be pathogenic? Is a question we were answering. And the answer to that is yes. We just published a paper a few months ago, entitled Characterization of Water Treatment Resistant and Multi-Drug Resistant Urinary Pathogenic E. coli in Treated Wastewater. And what we did here is we looked at these 50% that were not the naturalized forms, and we went in and we looked at their genomes, and we looked at their DNA, and we sequenced their entire genome of 5.2 million base pairs of DNA, and we interrogated the genome and said, what is it about these organisms? Are they pathogenic? And the findings were startling. So the strains that were coming out of a wastewater treatment plant were virtually identical to clinical strains causing urinary tract infections in humans. some of these strains were similar to clinical strains causing bloodstream infections in humans, and some were even similar to those that we know cause meningitis. And how we identified some of those is that those that cause meningitis carry a specific set of genes that allow them to cross the blood-brain barrier. And then we know these genes are important in their ability to cross the blood-brain barrier. And we were finding these genes in these particular E. coli coming out of a wastewater treatment plant, and for which they carried a number of other important virulence genes or disease-causing genes. For example, in one strain, we had 81 virulence genes, and these were identical in composition to pathogenic clinical strains that we knew, that we have examples of. Not only that, but they also carried an abundance of antibiotic resistance genes, with various strains clinically resistant to many antibiotics. We went in and tested the antibiotic resistance of these, and we found that many of them were multi-drug resistant. Not only were they multi-drug resistant, but they were multi-generational and multi-class resistance, a wide range of classes of antibiotics. And one strain was clinically resistant to 19 antibiotics out of 27 that we tested it against. And startlingly, many of the strains of these strains were considered to be of global pandemic concern. So for example, there's a particular strain of pathogenic E. coli that's circulating that we find across the globe, and it's called an extended spectrum beta-lactamase ST131 strain. Kind of a nomenclature we use not unlike COVID-19 to explain a complex thing. And this particular strain we know is across the globe and is infecting people, and we don't know where it's coming from. In fact, the European Union has commissioned a project looking at these particular strains and the potential role that water might play in the transmission, and our data suggests that it might be coming and being selected for in wastewater treatment plants for discharge. So if we go back to this question and ask, are these microbes pathogenic? The answer is yes. It seems that about 50% of the E. coli that's being discharged from a wastewater treatment plant may in fact be highly pathogenic, which is a startling finding that we made. And in fact, that the types of infections that they're causing are very unique, things like urinary tract infections or potential bloodstream infections. Or, as we're still trying to evaluate and have a paper soon to come out to talk about those strains that might be similar to those causing meningitis. This is a little bit even more problematic because as a trained immunologist and microbiologist, we have to understand something about the immune system as well. And have you ever wondered why that pimple on your face can sometimes be green? Or perhaps you've had a sore, a pussy sore, and why the pus is oftentimes green? Well, your immune system actually produces a number of highly toxic molecules to destroy pathogens as well. And one of the molecules that it produces is chlorine. So it produces hypochlorous acid or bleach at the site. And the reason that that pimple or that sore is green is because your white blood cells are producing chlorine or hypochlorous acid. The other thing that your immune system produces is a very potent advanced oxidants such as superoxide and hydroxyl radicals, peroxynitrate, nitric oxide, and peroxide as toxic molecules to destroy bacteria, viruses, or worms. And the question really becomes, could we be unintentionally creating the ultimate superbug? You see, to catch a criminal, you need to think like a criminal. And so in order to catch a superbug, you need to think like a superbug. So if I was a superbug, what characteristics would I want to have? Well, I would like to have a transmission medium that can effectively allow me to infect a large number of naive hosts. A single drinking water treatment plant can distribute water to 500,000 residents per day. Water is a very effective means of transmission. If I were an ultimate superbug, I'd want to develop resistance to the primary public health interventions used to control me, as Monica's already introduced us to. The intervention of filtration and chlorination in drinking water supplies has had a major role as a public health barrier. So if I was a superbug, I'd need to develop resistance to those primary public health interventions. And you can see from our data that it seems like some of them have. If I was a superbug, I'd want to develop resistance to clinical therapies aimed at killing me. And as I've already demonstrated and talked to you about, we do have antimicrobial resistance as being a major issue. And if I was a superbug, it would be great if the resistant mechanisms I develop were also effective at circumventing the immune system, which is the final mechanism of control. You see, in the water industry, we like to think we're pretty smart, that, you know, we invented chlorine, that we invented ozone and oxygen to kill microbes from. But it's important to understand that the immune system developed that millions of years ago, and that pathogens have been in a long fight with evolving resistance mechanisms to those very things that control. And so although we're using it in the water industry, understand that pathogens have had to deal with this for a long time. So if I was a superbug, I'd want to develop the resistance mechanisms against the very things that are intended to destroy me. And lastly, if I was a superbug, I'd disguise myself. So in other words, rather than cause diarrhea, as we suspect whenever I say the word waterborne disease, what's the first thing that pops into your mind? Well, probably vomiting diarrhea. So in other words, rather than cause diarrhea, which everyone would expect me to do, cause a disease that no one would expect me to cause. So if I cause a bloodstream infection, would the field really understand that it could be transmitted by water? So as you can see, if I was a superbug, I'd meet all the 5 kind of criteria that we already have in terms of the bugs that we're seeing starting to evolve during water treatment. So the potential for a superbug exists, but is it a problem yet? We've been able to describe that we think that some of the E. coli that are coming out of a wastewater treatment plant are actually highly pathogenic. But they may be, but we aren't quite sure yet. We have demonstrated that E. coli have evolved to survive and exploit the water treatment process, and that highly pathogenic forms are surviving this treatment process. And it is extremely worrying. But we also still see microbial reduction across the treatment train, suggesting that we may have an opportunity to alter our treatment process to increase killing. If we bumped up our treatment, could we actually kill these things? Is there a threshold at which they can no longer handle or evolve resistance to? What might that threshold be? Should we be moving to that threshold now? We need to consider multi-barriers. Each barrier is a hurdle for developing resistance, albeit, as I've already told you, the bacteria have developed resistance mechanisms against all major hurdles, such as chlorine, advanced oxidants, UV, microbial competition, and predation, and I would go so far as to say even ultrafiltration. And I can discuss that at a different time. And then the real question becomes, are the numbers released by our wastewater treatment plants sufficient for causing disease? What is the dose needed to cause infection? We think that the co-selection associated with the immune system will drive the infectious dose down of these things. So we fully anticipate that the infectious dose is probably quite low for these microbes, but we don't know. Is it 1 bacteria or is it 1 million bacteria? Because if a wastewater treatment plant is putting out 100 per 100 mLs, yet the infectious dose is 1 million bacteria, is it really a risk? We don't know those answers yet. And we're trying to develop animal models of disease to really understand this. And how confident would we be in these numbers? And these are the questions we're trying to answer with the funding from Alberta Innovates. And as you can imagine, have enormous implications for determining whether we're on the cusp of a public health crisis or not. So with that, I'd like to end and acknowledge the original project team members on this grant from the University of Alberta, University of Calgary. A lot of the work was done by my former PhD student, Dr. Shui Ji, who's now in China at Ningbo University in an academic position there, and we continue to collaborate on this topic. Norma Ricker from the city of Calgary, and Dr. Phyllis Posey, who used to be at Atlantium Technologies. And so with that, I will end.
Thank you, Norm. As always, I'm a mixture of fascinated and concerned after hearing your talk. Some really important information, I think, to how pathogens in our water systems might be changing and how we might need to be responding. We will move into the Q&A portion of the session. We've got about 20 minutes left for that. Please continue to submit your questions as we go through the discussion. I'll get to as many as we can. I want to start with just a general question for both of you. Based on the topic and the details that you've given, do you feel that we're at a point in time where we need to be reevaluating our water and wastewater treatment processes to adapt to these changes, or are we able to continue to use what we've used for a long time now, and make small modifications? Well, I'll start with Monica and give Norma a break here.
Thanks for the question, Vicky. So I think you pose a good question. In brief, yes, our treatment technologies work. We have evidence that they work. That's not to say that they work for everything, and I think Norma's probably scared the socks off of most of us. So suffice it to say, we're going to be employed for a while. But joking aside, we always have to be looking ahead to the next threat, the next challenge, and those steps become incremental. You said, are they small changes or are they big changes? I think Norm has very well pointed out that we need to think about not only the treatments, but the constraints around treatment, including what are the specific challenges, the specific contaminants, the specific pathogens we want to treat. And so my biggest hope for us is that our regulatory structures allow us to be nimble and responsive, because treatment is increasingly challenging. We are increasingly able to measure, identify these challenges, pathogens, for example, and link them ultimately to public health threats. But I would point out that some of the words Norm spoke about at the end are incredibly important. Let's make sure that the presence of these organisms is actually linked to a health outcome. One of my biggest concerns is that sometimes we forego real health threats because of potential health threats that are not well described. And so I think that emphasizes the need for toxicology and evidence and a weighing process. And the way we achieve that is by having an opportunity to have continuing discussion like we're doing here, and to have projects that allow us to reach across and to be speculative and to investigate, to discover, but then also to come back and have a reasonable discussion that allows us to weigh these challenges and make adjustments. So one of the biggest things I think we can do, frankly, is not have highly prescriptive regulations that then limit or even stifle innovation and treatment responsiveness. And I think that part of the Water Innovation Program, frankly, fosters those partnerships, allows us to have these discussions, and allows us to work with the various stakeholders relevant to the discussion so that we can be as nimble and responsive as we can, given that we're dealing with real health challenges.
Thanks, Monica. Norm, do you have any thoughts?
Yeah, I think appreciate the comments that Monica's made. I think the biggest risk to water is complacency. And the assumption that everything is safe and everything is fine, and we actually don't need any more research, or we don't need any more interests. I mean, everything is fine. We have this perception that we're not suffering from disease, and therefore everything must be fine. And I think complacency is our biggest threat to safety. And if you think, for example, about how we've been able to deal with waterborne diseases as we typically understand them in the case of diarrhea, we also know that the leading cause of waterborne illness right now is actually things like Legionella, respiratory disease. And that's been happening since 2005, that respiratory disease. So things change. And a very important law of physics is Newton's third law of physics, where he says that to every action, there's an equal opposite reaction. And that's the same in biology. Every time we change nature or exploit a system in nature, nature will respond. So whether that's through wildfires and the frequency at which fires will be coming under climate change, whether that's the evolution of new threats and microbial resistance, and some of these threats can be quite large. I mean, the World Health Organization anticipates that infectious diseases by 2030 and 2050 will become the #1 killers of humankind again. And as Monica demonstrated from her rates of disease and rates of infectious diseases, we really brought infectious diseases under control way back in the 60s. And we've seen a shift in the diseases from infectious diseases to chronic diseases as being the killers of human. But the World Health Organization is anticipating that infectious diseases will once again reign supreme in 2050. And largely due to antibiotic resistance. But as you can tell, it's not just antibiotic resistance we have to worry about. It might be treatment-resistant pathogens that evolve and emerge. And so complacency in these things is critical. You know, we used to think antibiotics were our savior in 1963. They were the magic bullet, and there's lots of evidence in the scientific literature at that time that we thought we conquered infectious diseases. You know, all you have to do is read the World Health Organization's report in 2014 to realize that was once our savior in 1963 is now our demon. And that's an important lesson. Complacency is the biggest risk to public health. And we need innovation. We need different ways of thinking about these things and understanding them and how to deal with water treatment. It doesn't mean that the technologies that we currently have aren't suited. It just means we have to challenge those and understand the mechanisms and the theory behind it. And I think that's what's critical. And to be honest, to say this right out loud, is I think that's what makes the Water Innovation Program with Alberta Innovates so excellent, is that it goes just beyond that to understand the theory and idea behind some of these things and understands the importance of addressing these questions for our future. And so that's a testament, I think, to the Water Innovation Program in Alberta Innovates and their commitment to that, as you can see through the research that is being done.
Thank you, Norm. Both great comments. Importance to maintain flexibility of research, not think that we know have all the answers and know all the information. I'm going to jump to some more speaker-specific questions here. So the first one is for Monica. Sorry, just pushing it to the slide so you can all see it. How do we crack the nut of complacency regarding risks to sources of drinking water and our ability to treat water at affordable rates?
That is a great question, and thanks, Phil, for asking the question. I think this gets to some of the broader discussion that has been reflected in some of the questions, and certainly Norm has touched on aspects of this as well with respect to complacency and identification of risks. I would say that another aspect of complacency, just to reiterate it, and then to get to answering the question, is not to forget about existing risks, which are, of course, very real. Threats to inadequate disinfection of the pathogens that we can disinfect have led to very significant, if not tragic, outcomes. Here in Ontario, where I am, the Walkerton tragedy is a great example of that. So I think we have to be very careful in not throwing the baby out with the bathwater, so to speak, first and foremost. But now let's get beyond that and assume that we will remember not to do that. And so the question is, how do we crack the nut of complacency regarding risks? And I think that that's a big challenge because it requires a very diverse group of people to come to the table. It's a total privilege to be here today, to be speaking with someone like Norm, who's clearly investigating the mechanisms as he's discussed and identifying the emerging threats to our water supplies. And then for me, someone like me who's very interested in treatment processes and managing those risks, the opportunities to bring those people together are very important. I think an aspect of the discussion that is missing, frankly, is the notion of the delivery of safe water and our construction around training around it. Let's not forget that technology, the idea of new treatment technologies, there's this belief in the community that technology will save us. We just have to build the right widget. The new one, the one that, well, okay, maybe it's not heating, maybe it's not chlorine, but what will it be that will crack this nut, as you've said? And so I think that one of the biggest challenges there is not to be complacent with respect to please deliver the new technology. By far, the multi-barrier approach that we talk about with respect to the provision of public health is going to be a component of that. So it's what we do on the landscape, how we manage these threats from ever making their way into our water supplies. So taking Norm's example of wastewater treatment, well, should we be requiring more treatment of wastewater so that those organisms don't make their way into our supplies? Or alternatively, should the burden be on the drinking water treatment plants that then treat whatever water and distribute it? So I think it's discussions about the water cycle holistically, what we can learn collectively through the various parts of the process where the water passes through with respect to the specific issue of water. And recognize that all of these factors are interrelated. The example that I gave was, surface water, that's where recharge comes from to the ground. It's not like groundwater is magically safe. And it's about having that clear, holistic discussion. And so I come back to the nut of complacency comes from cracking that nut, comes from interactions such as this, discussions, and then a commitment to policies that are not prescriptive to technology, but rather to outcomes. And this is something we face in education. Let's not force people to teach these subjects. Let's make sure that students come out with a skill set. I think that same kind of outcomes-based approach is what the water industry is going to face in the future. And what will be enabled by that is various organizations being able to come together to address these challenges. So regulators, so consultants, so private industry that can hopefully create some of the sensors we need to better identify these challenges, as one example. And this kind of discussion facilitates those connections. So thanks for the question.
I think that's a great segue into this next question here. As a society, should we continue to rely on end-of-pipe solutions, or is there a way that we can control the source? We all poop, but does it make it more sense for us to encourage more source urine separation within our centralized system? And I think this is for both of you, so you can each take a stab at answering that, perhaps.
Go ahead, Monica. I'll get you to answer the first part of that.
I'm going to give what's going to sound like a cheeky answer, but it's not meant to be at all. I think this is a really important question. So the question again is, does it make more sense for us to encourage more source separation within our centralized systems? I'll start with the beauty of centralized systems is economies of scale. So it comes down to how much we're willing to pay for things, number one. And that is the reality of treatment, whether it be wastewater, drinking water, stormwater, or beyond water. When we get to distribution of something like water, centralized treatment brings down cost. So that's number one. And then the question becomes, to what extent is it reasonable to expect society to pay for treatment as opposed to the individual? And again, that comes down to what society wants, what we literally vote for, what kind of policies we set. So absolutely, control at the source is meaningful. And dare I say, part of the future of innovation so that we can manage the spread of disease, especially if we get to a point where infectious disease becomes the norm with respect to being a major cause of death again. That's a scary future, but we need to prepare to engage in that potential scenario. So yes, my answer is yes. I think we do have to focus on source separation and thinking about how we can integrate that into financial models that are considered optimal because of management of cost and economies of scale.
So yeah, I would concur. I think one of the interesting things about mass distribution of water is we understand in particular drinking water. I know Harvard economists back in about 2005 produced a paper in a journal called Demography where they looked at public health impacts in the 20th century, and they found that the mass distribution of water and the costs in terms of life saved in investment were 23-1, making clean drinking water and the distribution of clean drinking water not only the most important public health and sanitation, being the most important public health interventions for control of infectious disease, far more than antibiotics or vaccination or anything else, that it was the most cost-effective intervention for control of infectious diseases. And so we always have to remember that it's the most cost-effective means to control infectious disease circulation in society. And so in some ways, end-of-pipe solutions like that still offer some advantages. But at the same time, we do have to start looking at where sources of risk may occur, because end-of-pipe solutions also open up other avenues and opportunities for other pathogens to evolve. And as I've said, when you think about distribution systems, you have to start thinking about things like Legionella, Mycobacterium, and Pseudomonas infections as well too, which are significant causes of hospitalizations and disease as well too. When it comes to controlling sources, one thing that I think we need to start looking at in Canada is those high-risk sources in terms of wastes that get back into systems. So I know the US is looking at this and the potential that perhaps hospitals need to consider having wastewater treatment plants, and that we can target specific points within centralized systems to have more advanced treatment options for them to prevent the entry of these things coming into generalized services. So, these are all types of solutions that I think we need to start thinking about. If we think the organisms are evolving in resistance, and we're seeing evolution of certain infectious diseases within hospitals as well too, and that they may be some of the interesting stories, for example, that relate to this phenomena that we've talked about in E. coli, for example. We've been able to identify the mechanism by which these E. coli get resistant to water. And the resistance mechanism itself is a piece of DNA. It's about 16,000 base pairs in length, and it's called the locus of heat resistance. And that locus of heat resistance is what we found is what was making, or a significant element of which was making the bacteria resistant to chlorine. That locus of heat resistance was actually first discovered in Klebsiella strains that were circulating in hospitals in nosocomial infections, associated with pieces of instruments that we couldn't sanitize or put into an autoclave, but that we had to pasteurize in the hospitals. And these bugs developed resistance to these pasteurization processes in hospitals and became some of the biggest problems in surgery clinics because of that. And so we have areas where, and again, hospitals are a great example, where, for example, 20% of the entire population in a hospital can be immunocompromised. And it's a great way for pathogens to evolve, change, adapt in those systems. And that's why we have such problems controlling some infections in hospitals. And we get those spillover effects as they come into the sewage treatment plants, and we're expected to treat them, but they're resistant to the things that from the hospital. So, potentially looking at individualized treatment systems at points of high risk become quite important. And like I said, the US EPA and others are looking at this potentially in terms of requiring hospitals to have wastewater treatment plants with specialized treatment and the highest levels of treatment that we can achieve in there with zero discharges. So there's different ways to address the problems that arise like that. And so source separation, for example, in the sewage treatment plant might become important and things like that. And there might be other ways that we can do it. Nature is very efficient at destroying these things as well too. And we can learn a lot from nature and the processes in nature and the destruction of these agents as well too. So.
Vicki, if I may, to just jump in and say very briefly, I would just like to point out, building on this discussion, if we don't do these things, if we don't engage in these discussions and start planning ahead, and if we are complacent, it's critical to recognize that the equity gap between the haves and the have-nots will grow. The most threatened populations are the rural communities, the remote communities, the marginalized populations. We're already starting to see that, not in some developing country, we're seeing that in North America, that the costs of treatment are adequate treatment are becoming prohibitive for increasingly larger fractions of society. And with that will ultimately come a price tag to society if we don't act to mitigate those risks. And so I just want to emphasize that all things are not equal depending on where you are in your socioeconomic status. And it is incumbent upon us as a society, because as we have seen, there is a domino effect of these illnesses in society. And so we need to look at it from that perspective as well, as opposed to just what goes on in our immediate backyards.
Thank you, Monica and Norm. That's a great way to end the session. We're at our time here. Unfortunately, a few more questions we weren't able to get to, but we do record the questions, and some of them we may be able to answer more directly to those that have asked them. Thank you again to our speakers. Great talks, a good discussion, some great insights I think that people can take back within their own organizations, and really looking forward to hearing some of the ideas that come out of this. I hope it has sparked some of those new ideas. A few people have asked. The session recording will be posted on our website in the next week or so. I posted a link to that website in the chat box. We will be sending out a short survey after the session. Your feedback will help us provide quality content as we continue through this monthly webcast series. There will be a draw for a gift card from those that respond to the survey, so extra bonus if you do respond. Our next session is our last session that is officially planned for, 2020 is ending. It'll be Digital Innovation Meets Water Management, held on December 10th. We are planning to continue some form of this series through 2021. We'll likely take a break at the start of the year and get going in February again. We're setting up some new topics and information. Keep an eye out on our website for what those sessions will be going forward. Thank you again, everybody. Appreciate your attending, and thanks to the speakers. Have a great day.